welcome to my channel Anupma Biology Classes. Today's topic is fertilization in reproduction series. This video is from chapter 3 part 6 of CBSE class 12th. Now in this video our first topic is fertilization. So fertilization is an internal process in case of mammals, and here the fusion of haploid male gamete and a haploid female gamete to form a diploid cell which is known as zygote, and this process is known as fertilization. It takes place usually in the ampulla of the fallopian tube. Now in the process of fertilization there are many steps, in which the first step is arrival of spermatozoa. Male discharges semen high up in the female's vagina close to the cervix during coitus, which is called insemination. From the vagina, the sperms reach the ampulla by two processes, in which the first one is sperm's effort. The sperms present in the semen travel a long way through the uterus into the fallopian tube by swimming in the fluid medium. And you know that sperm may swim at the rate of 1.5 to 3 mm per minute and at a single time of ejaculation. Sperms enters in the female vagina in 200 to 400 millions in number at a time. And in all these sperms, only approx 100 sperms can reach in the fallopian tube. Now the second is role of uterus in the sperm transport. Certain activities of the female reproductive tract helps the sperm reach the site of fertilization, like extension of the cervix into the vagina, aspirates the semen into the uterus from the vagina, and contraction of the uterus and fallopian tubes propel the sperms upward, and a sperm can reach in the fallopian tube in 5 minutes. But a sperm cell can survive in female's reproductive tract for 1 to 3 days, and it can fertilize the oocytes in 12 to 24 hours following ovulation. So this is the arrival of spermatozoa. Now here at first the sperm reach near the egg, then it attached to the egg. After this, in the process of fertilization, there are many steps of sperm entry and start fertilization, which we will discuss one by one, that how can it will be occur in the female tract. Now the arrival of egg. So the egg released from the mature Graafian follicle of an ovary is received by the nearby fallopian funnel and sent into the fallopian tube by the movements of fimbriae and their cilia. It is in the secondary oocyte stage with the second meiotic division in progress. The oocyte can live for 72 hours but can be fertilized only during the first 24 hours of this period. If copulation occurs at the appropriate time, the egg is surrounded by numerous sperms, and most of the sperms contribute enzymes which disrupt the follicular cells, still investing the eggs. Only one sperm succeeds in fertilizing the egg, others degenerate and engulfed by leukocyte. After this, capacitation of sperms. So the question is, what is capacitation? In this picture you can see the capacitated sperms release enzymes from their acrosomes in order to penetrate the cell and zona pellucida surrounding the egg. So the sperm in the female's genital tract are made capable of fertilizing the egg by secretion of the female genital tract, and these secretions alter the remove certain molecules deposited on the surface of the spermatozoa from the semen, and this process is called capacitation of sperms. It takes about 6 hours. Actually, sperms needs an alkaline medium to be motile, and in the secretion of the seminal vesicles, prostate glands and the Cowper's gland present in the semen contains nutrient which activates the sperm and substances that neutralize the acidity in vagina. So capacitation is important to make sperm active for the fertilizing the egg. Now the chemical and physical events of fertilization, in which there are 5 processes in these events. First is the acrosome reaction, then the cortical reaction, then sperm entry, then karyogamy, then activation of egg. So, in this process, at first the acrosome reaction occurs, in which a capacitated sperm passes through the corona radiata, which is the outer membrane of oocyte, you know that, to reach the zona pellucida, which is the inner membrane of oocyte. So, there are three glycoproteins are present in zona pellucida, in which the ZP3 function as a sperm receptor to bind to a complementary molecules on sperm surface. This binding causes release of hydrolytic enzymes by induction in sperm head, which are collectively called sperm lysines, and here calcium ion plays an important role in the acrosomal reaction. Now, the plasma membrane of egg fuses with the egg plasma membrane, and because of this binding, it induces depolarization of the egg plasma membrane and causes fast block of polyspermy that checks additional sperms from joining the egg plasma membrane. So this is the first block of polyspermy. After this, the fusion, here the egg shows cortical reaction to further check the entry of more sperms into the egg, and here the cortical granules, which is present beneath the X plasma membrane, fuse with the plasma membrane and release their contents between the plasma membrane and zona pellucida. This causes sure block of polyspermy. Then sperm's entry occur and the egg form the fertilization cone, which takes the entire sperm into egg. After this, the karyogamy occurs, and here the sperm entry stimulates the egg to resume and complete the suspended meiosis second. At this stage, the head of the spermatozoa, which has nucleus, separates from the middle piece and the tail and becomes the male pronucleus. And in ovum, female pronucleus present, and here the mixing of the chromosomes of sperms and ova occur, which is called karyogamy or amphimixis. This process completes the act of fertilization, and after this, the ovum is in diploid condition and form zygote, and the mother is said to be pregnant. Then the activation of egg starts within minute, and the rate of cellular respiration and the protein synthesis increase greatly. Usually, the sperms fertilize an egg within 30 minutes of ejaculation. So, this is the stages of sperm entry into the ovum during fertilization. Here, in this picture, you can see the complete process of acrosomal reaction, and in here, the contact occur of the sperm to the X membrane, then the acrosomal reaction, then growth of acrosomal process, then fusion of plasma membrane, then entry of sperm nucleus, then cortical reaction occur. So this is the complete process. Now, the sex of the offsprings. So, in human beings, each diploid cell has 46 chromosomes, 22 pair autosomes and 1 pair sex chromosome. Human females are homomorphic, while the human males are heteromorphic, because in the human female XX double X chromosomes present, while in case of male X and Y chromosome present, which are the sex chromosomes. The sex of the child is determined at the time of fertilization when female and male gametes fuse to form zygote. See in the picture here. Here the sex determination in an offspring by the parents, that is the father and mother, which have 46 chromosomes. In father, there is 44 autosomes and 1 sex chromosome, that is X and Y. While in mother, there is 44 autosomes and 2 sex chromosomes, that is X and X. So, at the time of gamete formation, they show meiosis and form 22 plus X and 22 plus Y in case of father, and 22 plus X and 22 plus X in case of mother. So, after the gamete formation, fertilization starts and one gamete make contact with the other gamete of the different sex. and form zygote, which have half chromosomes from father and half chromosomes from mother. So you can see here in the offspring formation there is chances of male or female child is 50-50%. So if a sperm carrying X chromosome fertilize an egg carrying X chromosome, then female offspring form. And if the sperm carrying Y chromosome fertilize an egg carrying X chromosome, the male offspring will be formed. So in future, male child or female child will born totally depends on the male chromosome because they are heteromorphic. Never on the female chromosomes because they are always homomorphic. So this is the sex determination in case of the offsprings. Now, what is the significance of fertilization? So, it provides stimulus for the egg to complete its maturation. It activates ovum to develop into a new individual by repeated mitotic divisions. It makes the egg more active metabolically. It combines characters of the two parents. It determines the sex of the young one to be developed from the zygote in humans. Ulemas, depolarization and hardening of zona pellucida resulting after sperm entry check the entry of additional sperms. Fertilization introduces centrioles which are missing in the ovum. It restores the diploid number of chromosomes in the zygote by adding male's haploid set of chromosomes. So these are the significance of the fertilization. After this, you can see here the species specificity in fertilization. And in this, a fertilizin is a proteoglycan secreted from the cortical region of the egg cytoplasm that interact with the anti-fertilizin of the sperm of the same species. This makes the sperms stick to the surface of egg. And anti-fertilizin is a protein present in the plasma membrane of the sperm, and here the fertilizin acts as a receptor for anti-fertilizin, and the reaction between the fertilizin and anti-fertilizin makes the sperm more capable of fertilizing the egg of the same species. In this picture, sperm contact with ova just because of the process of agglutination and interaction of fertilizin and anti-fertilizin. Because of this reaction, the sperms adhere to the egg. Now, the significance of fertilizin and anti-fertilizin reaction. It preserves the sperms. It ensures that at least few spermatozoa encounter the egg. It mutually attracts the gametes of opposite sex, and it ensures fusion of gametes of the same species. So this is the species specificity in case of fertilization because of fertilizin and anti-fertilizin, and fertilizin present in case of female and anti-fertilizin present in case of male. Now, the cleavage or segmentation. It refers to a series of rapid mitotic divisions of the zygote following fertilization, forming a many-celled blastula. In case of cleavage in human zygote, it occurs during its passage through the fallopian tube to the uterus. It is holoblastic. It means it divides the zygote and the blastomeres completely into daughter cells. The first cleavage takes place about 30 hours after fertilization. It is the meridional, coinciding with the animal and vegetal pole axis. Here, meridional means when a furrow bisects both the poles of the egg passing through the median axis or center of the axis. And the last is cleavage is radial and indeterminate in case of human zygote. So radial means blastomeres are arranged in radial plane around the polar axis, and indeterminate means the fate of each blastomere is not predetermined. So this is the starting of the cleavage. Now the first you can see here the stages of cleavage. And here in the first, cleavage takes place about 30 hours after fertilization and forms two blastomeres in which one is slightly larger than the other and adhere to each other. Then Second cleavage starts within 60 hours after fertilization and it occurs at right angle to the plane of the first and divides each blastomere into two by forming a mitotic spindle in each. And here, in this case, the larger blastomere divides a little sooner than the smaller one. So at first, 3 cell stage formed, then the 4 cell stage formed. After this, then the third cleavage occur about 72 hours of fertilization, which produces a solid ball of a small blastomere which is called morula, and it refers to the lobed surface of the embryo at this stage. So, in this stages, radial cleavage at 8 cell stage. Because of this, 4 cells are directly above the lower 4 cells and in indeterminate also, so each blastomere formed by early cleavage divisions retains the capacity to develop a complete embryo. Then it forms blastula where the outer trophoblast cells and the inner cells present, which we will discuss in the next slide. Now, the significance of cleavage. So, in case of significance, it restores the cell size and nucleocytoplasmic ratio characteristics of the species. It doesn't result in growth, though it increases cell number. Besides producing large number of cells by rapid divisions, also segregates different substances present in the cytoplasm into different cells. It produces a large number of cells so that the building of the offspring's body may begin. During cleavage, cellular activity is still mainly controlled by the organelles and the molecules received from the secondary oocyte cytoplasm, but some of the developing organism genes become active. Now, here the difference between the cleavage and the typical mitosis. So, in case of cleavage, it occurs in the zygote or the parthenogenetic egg, while the typical mitosis, it occurs in most body cells. In cleavage, interphase is short and the blastomeres do not grow during it, while in the typical mitosis, interphase is short and the daughter cells grow to normal size during it. In cleavage, the size of blastomeres decrease as their number increases, while in the typical mitosis, the size of daughter cells remains nearly constant due to growth. In cleavage, the total mass of the blastomeres remains the same as that of the zygote, while in typical mitosis, the total mass of the daughter cells becomes more than that of the parent cell. In cleavage, DNA synthesis occurs much faster to form chromosomes for new blastomeres, while in the typical mitosis, DNA synthesis occurs at the normal rate. In the cleavage, the nuclear cytoplasmic ratio increases as cleavage progresses, while in the typical mitosis, nuclear cytoplasmic ratio doesn't change. So this is the main differences between the cleavage and the typical mitosis. Now, morula in case of cleavage. Here in morula, the morula consists of 16 to 32 cells, but it is not larger than the zygote. Its cells are of two types. There is an outer layer of smaller, clearer cells around an inner mass of larger cells. In this picture, you can see here the two-celled stage converted in 16-celled stage, that is morula, which reaches the uterus about 4 to 6 days after fertilization. It is still surrounded by the zona pellucida that prevents it sticking to the uterine wall. Now, after the morula is converted into the blastula or the blastocyst. This is the formation of blastocyst. The outer layer of cells of the morula now absorb the nutritive fluid secreted by the uterine mucous membrane called trophoblast or trophoectoderm. In this picture you can see here, this is the outer layer of cells which forms the trophoblast. Now the fluid absorbed by the trophoblast collects in the cavity, that is the blastocele or blastocyst cavity. And this cavity separates the inner trophoblast from the inner cell mass except on one side. So, as the quantity of fluid increases, the morula enlarges rapidly and assumes the shape of a cyst. It is called blastocyst. Now, the inner cell look like a small knob at a pole. Here you can see this is the inner mass, which is look like a mass which is present on the pole, which is known as the embryonal knob, and on which pole embryonal knob present, which is called embryonic or animal pole, and the opposite end is known as abembryonic pole. Now, the trophoblast give rise to extraembryonic membrane, that is chorion and amnion, for the protection and nourishment of the embryo, and the trophoblast cell which connect with the embryonal knob are called cells of Rauber. After this formation of the blastocyst, zona pellucida disappears slightly. So this is the blastula. Now the process of implantation. In this, it is the attachment of the blastocyst to the uterine wall, and it takes place about 7 days after fertilization. Now, in this picture you can see at first after ovulation, the oocyte enters into the fallopian tube, and here the sperms present for the fertilization, and here the fertilization occur. Then at day first cleavage occur, then day by day 2 celled, 4 cell, 8 cell, and after this the 8 cell uncompacted morula, then 8 cell compacted morula form, then the early blastocyst form, after that the late stage of blastocyst form after hatching, then the implantation of the blastocyst on the uterine wall. So, the implantation involves activities of both the blastocyst and the uterus, and the embedded villi form villi around to obtain nourishment. And here it is established a parasitic relationship between the embryo and the uterus, which is called mother, and here the embryo protects into the uterine cavity and loses villi from the free surface. So this is the implantation. After this, now the role of zona pellucida. It prevents the implantation of the blastocyst at an abnormal site. It doesn't expose the sticky and phagocytic trophoblast cells till the blastocyst reaches the proper implantation site. Then miscarriage. What is the miscarriage? If the embryo is abnormal or dies, secretion of the HCG stops, and this lowers the progesterone output by the corpus luteum. This is in turn causes breakdown of the endometrium and a consequent miscarriage or abortion, that is loss of embryo. It commonly occurs during the first month of the pregnancy. Now, the human chorionic gonadotropin. The chorionic cells secrete a hormone called HCG, which resembles and takes over the job of pituitary LH during the pregnancy. HCG maintains the corpus luteum and stimulates it to secrete progesterone. The progesterone maintains the endometrial lining and causes it to grow throughout pregnancy, and this prevents menstruation. Now, what is the morning sickness in case of pregnancy? Implantation is also accompanied by certain other hormonal changes which causes growth and tenderness of the breast and temporary occurrence of morning sickness. Here, a desire of unusual food may appear as a part of behavioral pattern of pregnancy, and a pregnancy test or the woman's womb or blood test that is at present in the urine, which confirms the conception by showing the small amount of HCG. So, presence of HCG in the urine confirms that lady is pregnant or not. Now, after implantation, gastrulation occurs or the formation of germ layer. Here, in this picture, you can see the transformation of the blastula into the gastrula with 3 primary germ layers by the rearrangement of the cells is called gastrulation. and the germ layers are formed by the characteristic movements of some of the cells at or near the surface of the embryonal knob to their new, more interior locations, and these movements are called morphogenetic movements. So, this movement is accompanied by changes in cell shape and changes in the cellular adhesion to other cells. So, this is the start of gastrulation. In this picture, you can see how the blastocyst invades the endometrium and embedded in the uterine wall. If a blastocyst does implant, then the endometrium remains in the uterus and together with the uterine tissue becomes the maternal portion of the placenta called the decidus. So, the trophoblast cells then begins to intrude between the endometrial cells attaching the blastocyst to the uterine surface. So, the implanting gives the blastocyst a blood supply so that it can start growing into a fetus. Along with the cramping, you may experience what is called implantation, bleeding, or spotting, and this usually happens 10 to 14 days after conception, around the time of your usual period. So, there are three germ layers, ecto, meso, and endoderm, and they serve as the initial rudiments of all the future tissues and organs of the offspring's body. Each germ layers give rise to specific tissues, organs, and organ system of the young ones to be developed. Now, how the endo, meso, and ectoderm form? So, some cells covering the free surface of the embryonal knob differentiate, they become flat and form the first endodermal cells. So, some cells covering the free surface of the embryonic knob differentiate and then they rapidly multiply and spread out in all directions and soon form a complete lining inside the trophoblast of the blastodermic vesicle. This lining constitutes the endoderm. Now, the formation of embryonic disc and the amniotic cavity, then we will know how to form ecto and mesoderm. So, the blastocysts grow in size by absorbing uterine fluid through the trophoblast. With the growth of blastocyst, after the formation of endoderm, the embryonal knob is stretched and its cells become columnar and come to lie as a regular layer which is called embryonic disc. Then a space appears between the embryonal disc and the trophoblast, which is called amniotic cavity, which is filled with amniotic fluid. After this, at the hind end of the embryonic disc, the cell starts proliferate at increased rate, and this proliferated cells later detached from the embryonic disc and is spread between the trophoblast and the endoderm. Then the new layer of cells are formed which is known as the mesoderm, and it separates the yolk sac and the amniotic cavity from the trophoblast. After the formation of mesoderm, the remaining cells of the embryonic disc get arranged in a layer which is known as the ectoderm. So this is the formation of endoderm, mesoderm, ectoderm, and the formation of embryonic disc and the amniotic cavity. Now, the formation of extraembryonic coelom. A large cavity, the extraembryonic coelom, is formed in the extraembryonic mesoderm. The latter is split by this activity into two layers, that is the parietal extraembryonic mesoderm and the visceral extraembryonic mesoderm. So in this picture you can see now the developing embryo along with the amniotic cavity and yolk sac is now suspended in the extraembryonic coelom and is attached to the trophoblast by the upsplit part of the extraembryonic mesoderm. With the formation of extraembryonic mesoderm and the extraembryonic coelom, the primary yolk sac becomes smaller and comes to have a wall of endoderm and mesoderm. It is now called the secondary yolk sac. So this is the formation of extraembryonic coelom. Now, the process of organogenesis. In this process, the establishment of the germ layer initiates the final phase of the embryonic development, that is called organogenesis. It involves differentiation and specialization of the groups of cells in the individual germ layers. The primordial cells of the germ layer gradually and accurately give rise to the tissues and organs of the offspring. Then, in human beings, by 4 weeks after fertilization, the embryo develops a simple heart. By the second month of pregnancy, limbs, buds, and the eye rudiments of the fetus develop. By the end of the first trimester, most of the major organ systems are formed. From this stage, embryo is called fetus. The first movement of the fetus and the appearance of hair on the head are observed during the 5th month of pregnancy. So, by the end of second trimester, that is 24 weeks of pregnancy, the body of the fetus is covered with fine hairs, eyelid, separate eyelashes which are formed. Then the fetus becomes fully developed and ready for delivery by the end of 9 months of the pregnancy. So this is the formation of the organs in case of the embryo, that means in the fetus. Now, the fate of germ layers, in which you know that there are three germ layers: ectoderm, mesoderm, and endoderms, and they all are used in different parts formation. So, the first one is ectoderm, and in this it gives rise to, that is, epidermis of the skin and the epidermal derivatives, nervous system, medulla of adrenal gland, posterior and anterior lobe of pituitary gland, pineal gland, eye, internal ear, nasal epithelium, lining of stomodium and proctodium, lining of external auditory canal, lacrimal glands and salivary glands, enamel of teeth. So these all are found by the ectoderms. Now the second is mesoderm, and here it produces the dermis of the skin, most muscles, connective tissues, peritoneum, kidneys, cortex of adrenal glands, gonads, urinary and reproductive drugs, heart, blood, lymph vessels, spleen, wall of the gut except its lining epithelium which are formed by the epidermis, pericardium and pleura, sclera and choroid of eye, most of the skeleton, dentine of teeth, mesenteries, notochord, all these structures are formed by the mesoderm. Now, the third, that is the endoderm. And endoderm forms the lining of the gut except that of the stomodium and the proctodium, gastric and intestinal glands, tongue epithelium, liver, pancreas, anterior lobe of pituitary gland, thyroid gland, parathyroid gland, thymus gland, etc. Then lining of trachea, bronchi and lungs, lining of urinary bladder, lining of middle ear, primordial germ cells and the greater part of prostate. All these are formed by the endoderm. So these all are the fate of germ layers. Now the extraembryonic or the fetal membrane. So this is one of the membrane which assist in the development of the embryo, and such membranes occur in a range of animals from humans to insect. And here you can see that there are four fetal membranes, in which the first, like the yolk sac, amnion, allantois and chorion, these all four are the fetal membrane which we will discuss in the next slide. So here you can see here that all the embryonic membranes, that is extraembryonic membranes, that originate from the embryo but are not considered the part of it, and they are typically perform the role in nutrition, gaseous exchange and the waste removal. So here now the first, that is the yolk sac, and it is formed below the embryo and contains fluid, not yolk. So because we are alacithal, and it functions as the site of blood cell formation until about the 6th week when the liver takes over this role. Thereafter, the yolk sac starts to shrink Its wall consists of endodermis and the mesodermis. After this, the second is amnion. It is formed above the embryo and it consists of trophoblast inside and the mesoderm outside. It closes a fluid filled space that is known as the amniotic cavity, in which the amniotic fluid present and have many functions, like it cushions the embryo, protecting it against bumps and against pressure of maternal organs. It permits fetal movements, prevent bacterial infection, maintains a constant temperature and pressure, protects the embryo from the injury if the mothers fall, etc. So this is the work of the amniotic fluid which is present in the amniotic cavity in amnion. Now, the third is allantois, and it is a sac-like structure and pushes out from the gut of the embryo near the yolk sac by the third week, and it consists of endoderm and mesoderm, and it helps the embryonic exchange of the gases and handle the liquid waste. And the last is chorion, which surrounds the embryo and it is composed of trophoblast and mesoderm, and it protects the embryo from the placenta for the metabolic exchange between the fetus and the mother. So, after implantation, endometrium undergoes profound changes and gets modified. And this modified endometrium of the uterus is called decidus stage, which you can see in this picture. This is the decidus stage. Now, here you can see the placenta. So, what is placenta? In this picture, you can see this is the structure of placenta, and it serves to bring the fetal and maternal blood close enough to permit the exchange of materials between the two. And in the picture, you can see it is reddish brown disc about the size of a dinner plate and little less than 1 kg in weight. And in this picture, you can see the placental components like the amnion, fetal vein, fetal artery, maternal blood, decidus basilis, maternal veins, bronchus villus, okay, and syntotrophoblast, cytotrophoblasts, etc. So these are the placental components. Now, here, a large number of branching villi from the vascular chorion penetrate the corresponding pits, that is, the crypts, formed in the uterine wall. Then, the intimate connection established between the fetal membrane and the uterine wall is hemochorial placentation. And the placenta has two parts. First one is the fetal placenta, and next is the maternal placenta. So, the cells forming the walls of the chorionic villi bear microvilli which increases their surface area for the adequate exchange of materials by diffusion, active transport and pinocytosis. So this is the attachment of the fetal placenta to the maternal placenta. Now here you can see the nutrient exchange, and for this in the placenta the fetal blood comes very close to the maternal blood, and this permits the exchange of material between the two. Food like glucose, amino acid, proteins, lipids, water, mineral salts, vitamins, hormones, antibodies and oxygen passes from the here the maternal blood into the fetal blood, and the fetal metabolic wastes like CO2 and urea and also hormones and water passes into the maternal blood. So the placenta serves as the nutritive respiratory and excretory organ for the fetus. Now, the function of placenta. The first function is nutritive organ because the food material passes from the mother's blood into the fetal blood through placenta. The trophoblast of the placenta digest protein before passing them into the fetal blood. Gaseous diffusion between the maternal and fetal blood through the placenta. Nitrogenous waste from the fetal blood into the maternal blood via placenta for the elimination by the mother's kidney. It secretes HCG, estrogen, progesterone, and the human placental lactogen. It stores glycogen for the fetus before liver is formed. And the last is barrier. It acts as a barrier and many substances pass from the mother to the fetus via placenta and causes abnormal development in developing embryonal fetus. Such substances are called teratogens. So these are the functions of placenta. Now, types of placenta, in which the first one is epitheliochorial. In this, the chorion is next to the lining of the uterus but doesn't invade or erode the lining. Second is syndesmochorial. In this, the epithelium of the maternal tissue is eroded to facilitate intercommunication. Next is endotheliocorial. In this, the syncytial trophoblasts of the chorion penetrate to the blood vessels of the uterus. Next is hemochorial. In this, the maternal blood is in direct contact with the chorion. Next is endothelial placenta, and here there is no trophoblast and epithelium, they are in the direct contact. And the last is hemoendothelial. In this, the placenta having the endothelium of the vessels of the chorionic villi is direct contact with the maternal blood. So these are the types of placenta. Now the parturition. It is the act of expelling the full-term young one from the mother's uterus at the end of gestation, and the gestation is completed in about 20 to 80 days from the start of mother's last menstruation. In this picture, you can see here the hormonal induction of labor. Here, the fetus signals that the baby is matured by secreting certain hormones. Here, the estrogen from the ovaries induces oxytocin receptors on the uterus, which stimulates the uterine contractions, and this provides force to expel the baby from the uterus, causing birth. It also stimulates the placenta to secrete prostaglandins, which stimulates the contraction of uterus. And here the uterine contraction by positive feedback induce continuation of oxytocin and prostaglandin production. So this is the positive feedback and hormonal induction of the labor. Now, what is labor? So, the labor is the forceful muscular contraction of the uterine wall leading to parturition, which are called labor. Now, the stages of parturition. So, in this case, there are three stages. The first one is known as dilation, second is expulsion, and the third stage is known as placentation or placental, which you can see in the picture. So, after the 9-month-old fetus, then the first stage starts, which is known as the dilation. It lasts for about 12 hours, and regular peristaltic contraction of the uterine wall begins and passing from it top downward. And this contraction moves the fetus down towards the cervix, which expands fully. Now, the amnion and chorion rupture. The amniotic fluid passes out, lubricating the vagina, and the fetal placenta is pulled out from the uterine wall, making the young free. Then the next stage, that is the second stage of birth, which you can see in the picture, that is known as expulsion. It lasts for about 20 minutes to 1 hour. The uterine contraction become more powerful and are accompanied by contractions of the abdominal wall. With these contractions, the young one is gradually pushed outward through the dilated cervix and vagina with the head foremost and is finally delivered, that is born into the outside world where it draws its first breath. Here, the umbilical cord is ligatured at two places and close to the baby and cut between the ligatures. This frees baby totally from the mother. Cervix and vagina are dilated and the ligaments of the pelvic girdle are relaxed by the hormone relaxin from the corpus luteum for easy birth of the young one. After this, the third stage, that is known as placental, and is last for about 14 to 45 minutes after the infant birth. Here, the minutes after the infant birth, the uterus begins to shrink, and this frees the placenta from its implantation site. Now, the placenta, umbilical cord, and the fetal membrane are expelled by another series of uterine contractions, and these structures are referred to as afterbirth stages. So, these are the complete stages of the parturition. Now, the prevention of bleeding. Further, the contraction of the uterus closes the cervix and prevents excessive bleeding. After this, the normalization of uterus, it is about 28 to 35 days, and the uterus returns fully to its non-pregnant state by reduction in size and restoration of the endometrium. After this, the period which is known as the lactation. So in this period, the production of milk in the female's breast following the birth of a young one in mammals is called lactation. Composition of milk is proteins, fats, sugar, vitamins, mineral salts and water. It is poor in iron and has a very little amount of vitamin C. Then what is the benefits of milk? Milk can fulfill the entire requirements of the infant for growth and energy. Milk can produce a weight gain of 20 to 30 gram per day in the infants. So what is the colostrum? After the birth, the breast first releases not milk but the colostrum for 2 or 3 days. It transfers antibodies from the mother to the baby to combat infection. It is also called foremilk and it contains cells from the alveoli and is rich in protein but low in fat. Now, twins. So, what are twins? During evolution, the natural selection has strongly favored the birth of only one baby at a time in humans. A single baby tends to be larger and healthier with a better chance of survival. A mother can bring up and look after a single baby better and more easily. Sometimes one human female produces two babies at a time. This is called twinning. So one of the two young produces at the same birth is termed as twin. And here there are two modes of twinning. The first is from one egg and second is from two eggs. So, in case of from one egg, if the early blastomere separates before their fate is fixed, each can develop into a complete young one. And both the young one formed in this manner have the same genetic makeup as they have the genes of the same egg and the same sperm. And they are always of the same sex, have identical traits and are almost indistinguishable. Then they are called identical twins and they are now more appropriately known as one egg or monozygotic twins as they arise from a single egg. And the second is from two eggs. In this case, at times the two eggs, one from each ovary, may be released simultaneously and both are fertilized each by a separate sperm. Each zygote develops into a young one, and such young ones are genetically different because each receives genes from a separate egg and a separate sperm. They may be of the same or the different sexes. They are called non-identical or fraternal twins, and they are now called two-egg or dizygotic twins as they arise from the two zygote. So, here how twins are made. So in case of fraternal twins, two sperms fertilize two separate eggs which develops in womb at the same time. So the DNA 50% maternal and the 50% paternal. While in case of identical twins, the single sperms fertilize single egg which divides and makes two babies. So the DNA is 100% maternal and 100% paternal. And in case of semi-identical twins, two sperms fertilize one egg which divides and makes two babies. So in this case 100% maternal and 78% paternal cases. Now, in case of identical or monozygotic twins, one egg released from ovary and fertilized by one sperm. So here there are three cases. In the first case, early embryo splits before implanting in womb. And here the separate placentas and the separate inner sacs occur. While in the second, that is the early embryo implants in womb and then splits. Then the shared placenta and the separate inner sac. While in the third, the early embryo implants in womb and then splits later. In this case, shared placenta and shared inner sac occur. So these are the consequences in case of the identical twins formation. Now, in case of fraternal twins, it develops from two sets of egg and sperms and same sex or one of each. And in case of the identical twins, develop from same egg and sperm, and here same sex, or two males or the two females. So these are the differences between the fraternal and the identical twins. Now, the polyembryony. Regularly in armadillos and rarely in other mammals, including humans, a single fertilized egg may give rise to 4 or even 8 identical embryos, and this condition is called polyembryony. It is different from the position in the polyovulatory species that normally produce more than one ova per cycle, including rodents and dogs. Now, the developmental disorders. And in this case, there are three disorders. First one is amniotis. And in this case, the inflammation of amnion due to infection resulting from premature rupture of amnion often leads to neonatal infection. Second is abortion, escape of an embryo prior to the stage of viability. And then teratogeny. In this case, the production of malformed infant due to use of tobacco, alcohol and drugs by the pregnant mother. So this is the end of the video in which you understand about the fertilization process, implantation, extraembryonic membrane, organogenesis, fate of germ layer, embryonic membranes, placenta, parturition, lactation, twins, polyembryony and the developmental disorders. So the next upcoming video will be on the reproductive health. And if you understand this video, so please like and share my video and subscribe to my channel Anupma Biology Classes. If you have any queries, any questions, any suggestions, you can ask in the comment section below. Thank you for the watching.